Cheers, thanks. Hi, everyone, and thank you very much for the opportunity to present my work. So this presentation is about the work we have been doing in CSIRO for the past 3 years about sugarcane root phenotyping and the solution we developed to do rapid field phenotyping for sugarcanes. So for those who are not familiar with sugarcane, it is a tall tropical C4 grass. And as you can see on this picture, it can reach up to 4 meters. And in Australia, sugarcane is cultivated in the northern part of Australia, in Queensland. And it is a crop that is propagated vegetatively from set, as the one you can see on this picture. So a set is a piece of stalks that contain a bud that will germinate to become the primary shoot, and a round of root primordia that will become the set roots. So once the sets are planted into the field, the crop cycle is about 12 months until the plants are cut for harvesting. And after harvesting, the base of the plants remain in the soil, and the bud at the base of the plant will germinate again, and will become the next crop, also called ratoon. So usually in Australia, this cycle goes on for 3 to 5 years, until the yield loss are becoming too important and the farmer has to replant. So, as I mentioned before, when the set germinates, the first root to appear will be the set roots. They are fine, highly branched, but short-lived, and they are only there to sustain the first few weeks of bud germination and elongation. The second type of roots are the shoot roots, and they are thick, less branched, and they're making up the majority of the root system. And one thing to notice is that each stalks or tillers will have its own set of nodal roots. So the sugarcane root system, in addition to its role in water, nutrient uptake, and interaction with microorganisms, has a really important role to play in the plant anchorage to prevent the lodging of the plant due to the plant tall stature, and also to prevent uprooting at harvest. And the root system also plays a role in the ratoon vigor to help the next crop establish. So far, there haven't been a lot of studies about the sugarcane root system, and the current knowledge is that the maximum depth of the root system is around 4.2 to 4.7 meters, that 80% of the roots are located in the top 60 centimeters of the soil, and that the root dieback between ratoon cycle is around 17%. And these studies are great, but they do not provide any information about what the sugarcane root and root system looks like. And we need to have this information to help us build a sugarcane root system ideotype. So 3 years ago, we started our sugarcane roots project by conducting an initial screening of the root system of 20 sugarcane cultivars. And from this screening, we selected 5 sugarcane cultivars on which we study the effect of reduced tillering on the sugarcane root system. So in other species like wheat, reducing tillering either by genetic selection or by manual tiller removal leads to an increase of the root-shoot ratio. And we wanted to know if it was the case also for sugarcane. So in this experiment, plants were cultivated in 1 meter tall PVC pots under ideal conditions for 40 weeks, and half of the plants were able to grow freely, and the other half had their emerging tiller removed manually every single week. And at harvesting, amongst other things, we measure the stalks and the leaf mass fraction, the root system opening angle, and all the roots were scanned, dried, and weighed. So after 14 weeks, as you can see on this picture, the plants were well developed, but at this stage, it was too early for the sucrose to accumulate into the stalks, and so we could not measure the yield. And the root system of these plants were quite extensive, but not pot-bounded. And after scanning them all, the total root system per plant was between 900 meters to 1.7 kilometers. So Yes. That was long hours of scanning. So obviously, as you can see on this graph, the number of stalk was different between treatments. But while the restricted plant here in red had less stalk than the free tillering plant here in blue, their individual stalk weight was, in average, 85% greater compared to the free tillering plants. But still, overall, the total shoot dry weight, total shoot weight was 50% higher in the free tillering plants. And then when we look at the root system of these plants, we observed that the number of nodal roots was different between treatments, but just only in average by 40%. And this was due to a massive increase of the number of nodal roots per stalk in the restricted tillering plant, which had an average of 52 nodal roots per stalk, while in the free tillering plants, the number of roots was only 10. And then it was really interesting to see the impact that the number of nodal roots and a number of tillers had on the root system opening angle. And you can see an example of this on this picture, which were obtained by hanging the root system of this plant on a black box illuminated by LED. And then, as you can see on this graph, the root system opening angle were not that different between treatments. So while there was a massive reduction in tiller numbers between treatments, the root system opening angle of the restricted plant was only decreased by 10 degrees. So finally, to come back to our initial question, does restricting tillering increase the root mass fraction? As you can see on this graph, the answer is no. There is no significant effect of restricting tillering on the root mass fraction, and the trend is even toward a decrease in RMF in the restricted plants. So in this case, like sugarcane doesn't behave like wheat does. So later on, using the same setup, we conducted an experiment on the effect of nitrogen limitation on the sugarcane root system. And here we wanted to know if the sugarcane root system can adapt to limited N resources, and if there was any root system or root trait that can help reducing N leaching, because this is a massive environmental issue in Australia due to the proximity of the Great Barrier Reef. So for this experiment, plants were cultivated for 14 weeks, and half of the plants were treated with high nitrogen and the other half with low nitrogen. And I won't go too much into the detail of the general results, but first we observed that the low nitrogen plant had a higher root mass fraction, and this was expected. And we also show that the low nitrogen plant did not have a steeper root system opening angle, but they had nearly 50% of their root system volume located in the bottom 30 centimeters of the root system, while the high nitrogen plant had most of their root system volume in the top 60 centimeters of the root system. So it looks like the low nitrogen plants are investing more resources to go deeper. And so the next question was, is there any adaptative mechanism that helps the plant maintaining or increasing its root system size when the nitrogen resources are limited? And we tested this hypothesis by doing cross-section of nodal roots near the root tip and at the nodal root midpoint. And we measure several parameters relative to the root energy efficiency. And here you can see a representative image of the root cross-section. And first we observed that there were 1.5 fewer metaxylem vessels under the low nitrogen condition. And then when we look at the cortical cell numbers and their size, we did not observe any significant difference between treatments. So it looked like the nodal roots do not reduce their cortical burden when the nitrogen is limiting. But while the cortical burden was not different between treatments, we found it to be significantly different between variety. And here I'm showing you two extremes, so two variety, SRA1 and Q151. And as you can see, close to the root tip, SRA1 nodal root were larger compared to Q151. They had more metaxylem vessels and about 3 times more cortical cells than Q151. And then when we look at the cross-section made from the nodal root midpoint, this is what we saw. It was like parenchyma everywhere. And here I'm just showing you the example of Q151 and SR1, but it was the same for all variety in both treatments. So it looked like parenchyma is a constitutive feature of the sugarcane root system, and that the sugarcane root system tend to be energy efficient after all. So that's good so far. We had interesting result in the glasshouse study. But the issue with such big plants in glasshouse is we tend to be limited by space, and we can't grow the sugarcane long enough to be able to measure the sucrose yield. So we had to do some field work, but there are several issues with field work about the root recovery time, the fine root loss, and the heat, the mosquitoes, and you can have the crocodiles and the snake in Australia. But the number one enemy of doing phenotyping roots in sugarcane, it's the plant size itself. And this is the base of a 3-year-old sugarcane plant. And as you can see, it is massive and not practical to work with. So the question for us is how to do efficient field phenotyping. And this is where by chance encounter, we've been in touch with the South Australia Research and Development Institute, SARDI. And researchers at SARDI have developed a robust technique to extract and quantify DNA from a target species from soil sample weighing up to half a kilo. And the quantification is done by qPCR, targeting the internal transcribed spacer, ITS. So in partnership with SARDI, we developed this method to work with sugarcane. And here, this is a bit of background about the test development. So first, after making sure that our test was specific to sugarcane, we had to quantify the ITS. We quantified the ITS copy number for 31 sugarcane cultivar. And you need to do this because the ITS copy number could vary greatly between cultivars. So if you want to compare the result from a field experiment with involving multiple variety, you need to control for the ITS copy number. So as you can see on this graph, for sugarcane, the ITS copy number did not vary that much between cultivar, except for the 3 on the far right of the graph. And from this experiment, we've been able to calculate a scaling factor for each variety. So then, using 8 cultivar, we wanted to calibrate the test, yes, thanks, in order to have the result, which normally I express as target DNA quantity per gram of soil, and to convert the test result into root dry weight per gram of soil. And the results are presented here, and as you can see on this graph, the DNA test results were well correlated with the root dry weight. But it was interesting to see that for the lower part of the root system, which is the dot and line in red, was slightly more DNA dense than the top part of the root system, which is here in blue. And I have to say, it's plants that were cultivated in these tall, 1 meter tall PVC pots. So finally, we tested the ability of our method to detect only live roots. And to do this, we took moist soil and air-dry soil, and we mixed sugarcane roots with dry or moist soil. And then we follow the root DNA decay after 1, 3, 5, and 7 days. And as you can see, after 7 days, so there are the box blocked in blue, there is only 1% of the original DNA that remain, while in the air-dry soil, the DNA did not degrade. So at this stage, our method was ready, and so we took it for a test drive into the field, and we conducted a preliminary study on the effect, on the root dieback after harvest. And according to the literature, 83% of the root system remains alive after harvest and help with the establishment of the next crop. So to test this hypothesis or previous result, 12-month-old plants were from 4 cultivars were cut at T0, and soil core were collected around the bases of the plant at T0 and 1 week and 2 weeks after harvest. And the soil core were 60 centimeters deep. We split them in 15 centimeter segments, and to make one sample, we pulled 8 50 centimeter core from the same depth. And the results are presented here on this graph, where the y-axis represents the soil depth and the x-axis represents the relative root density. And as you can see, the decline in root mass was positively and significantly correlated with the depth of the soil core sample, whereas overall, time did not have a significant effect on the quantity of viable sugarcane roots, at least over this 2 weeks period. So this tells us that our test is working, and that these really preliminary studies, results, tend to agree with literature on the root dieback. So in conclusion, from our glasshouse experiment, we scan about 152 kilometers of roots, and we observe large sugarcane root system genotypic diversity and resilient root-root ratio and energy efficient root system. And from here, in glasshouse experiment, we can now focus on individual root rates, which are important for the specific constraint of the sugarcane cultivation, which are mainly about soil compaction and nitrogen leaching. And then to study the entire sugarcane root system and the root distribution in the soil, we can use our DNA method. And the method is working, but in order to get reliable result now, we need to determine how the core are collected, how many we need to collect, and where we collect them in order to get like a good, reliable result. And so we're also developing now a new calibration where we're trying to express the result as fine root surface area instead of the root dry weight. And on that note, I would like to acknowledge all the people from the CSIRO Sugarcane team that were there and helping with all the scanning, and people from SRA, Sugar Resource Australia, for the funding, and people from SARDI as well. Thank you very much.